Hi everyone, my name is Ryan and I'm going to be talking to you briefly about how to install Dana, DNA Area NETosis Analysis, on your Windows computer. Okay, so the first thing that we're going to do is we're going to get a hold of the basic components of Dana. And to do this, we're going to navigate to Dr. Shalef's website. I've linked it below in the comments, but I'll copy and paste here. And if you look over here along the left-hand side, there'll be a form to download Dana software. All you have to do for that is you have to fill out your first name, last name, degree, university, employer, and email. And when you do that, you can click Submit, and it'll take you to a link to download a zip file containing all the components you need. I already have a copy of this file right here. It's in this folder called Dana. And if I open it up, it's going to contain folders for the output, folders for the Fiji macros, the executable file for Dana, as well as other miscellaneous components. Okay, now that we have most of the Dana files, the next step is going to be to install Fiji and load the first part of Dana. You can readily do that by going to the Fiji website. You search ImageJ Fiji. It'll pop up right here, and then you can click Download. I'm going to assume that you've already downloaded Fiji. In fact, I have myself. Okay, so the first thing that we're going to do is we're going to edit the first part of Dana, Dana Part 1. Dana Part 1 operates within Fiji. It's actually a Fiji macro. And to do this, we need to change some things about the macros that we've given in this Fiji macros folder. So I'm going to highlight both of these and then drag them into Fiji. And that basically opens the files to allow us to edit the code. And the first thing that I'm going to do within this is I'm going to highlight this microscope calibration part. What this is for is that when you analyze your own images, the microscope distance and the micron distance are going to be essential for calculating correct areas of your regions of interest, or your ROIs. So in our case, 270 pixels in our microscope corresponded to 100 microns. So when you calibrate your own microscope, these values may be slightly different, but you should just change them within here and then proceed for further use. Additionally, what we need to change in this file is the location that Dana 1 is going to export its files to. And that's right down here. It shows your folder location goes here. And where I'm going to put mine, just for basic installation, is within this Dana 1 output folder that we saved in this Dana folder right here. And to do that, I'm going to copy the file path for it, and then I'm going to copy it, or paste it right here. And one small caveat about this is that there need to be two backspaces instead of one separating the locations. And when you complete this, you'll notice that it turns a pretty pink color. Okay, and you need two right on the end there to separate our file path from our newly created file. So I'm going to save this, and then copy this file path. It's going to be pasted in, if you look up here, we've opened two files. This was the first one for single use, and then we also have one for batch use. This follows the same process. Change your microscope scale if need be. Additionally, we're going to save or change our output location. And I'm going to paste that there. And then I'm going to file save these, and then I can exit out of this editor. The next thing that we want to do is we want to put those Fiji macro files, or the first part of Dana, within our Fiji plugin. And to do that, I'm going to exit out of Fiji, and then I'm going to go look at the files that I've opened, which are right here. And to do this next part, we have to find the location of our Fiji folder, like where the program is saved in our computer. I've saved mine to the desktop for convenience, but yours is probably in a different location. So what we're going to do is we're going to open this Fiji app file, and we're going to navigate to the plugins section, then the scripts section, then the plugins section again. And then we're going to take these two files that we just edited and paste them in this location. Okay, so now that that's done, we can reopen Fiji. So I'll actually open this back up. Open my Fiji whenever it decides to load. Okay, so my Fiji bar just opened up again. And now you'll notice that if you go to the plugins folder, Down towards the bottom here, we have two new macros, which are the Dana Part 1 and the Dana Part 1 batch. So those should contain our proper microscope calibration, as well as output to the proper location that we've selected. Okay, so now that we have the first part of Dana set up with an ImageJ, the next part is going to be to run Dana Part 2. If we open our Dana file folder, Dana Part 2 is just this executable file right here. To run that, we're just going to double click it. So this is Dana Part 2 right here. If you are having problems loading Dana Part 2, your version of Java may not be current. And to fix that, all you do is go to Safari, search Java, and then get the free Java download. Once you have that, you should have no problems getting this to open. Now that we've successfully installed Dana Part 1 into Fiji, we have our Dana 1 macros right here. And we are also able to open our executable file shown right here, available right here. We're going to end this video. Future videos are going to talk about how to optimize Dana to your specific sample or stain or cell type. And we're also going to talk about how to use Dana both in the regular mode and the batch processing mode. Thanks for listening.